We've been fortunate to have had a number of times in before. He's most known for that wonderful Darwin uh, sticker. Essentially found that the transitional species that I remember creationists telling me back in the 70s and 80s was missing. He's not just done that, but kept on not just teaching students, but looking around for other creatures of interest, mostly in the Devonian period. He's recently retired from the Academy of Natural Sciences after 30 years of building a great collection, research, and teaching. He had studied at Franklin and Marshall in our state of Pennsylvania, master's degree in paleontology from the University of Berkeley, and got his PhD from the University of Pennsylvania. So take it away, Ted, and thanks for being here today.
Thank you, Eric. Thanks for the nice introduction, and thanks everyone for coming around for this. It's really exciting for me to be able to share the research program that I have been up to for about 30 years. I did just retire from the Academy of Natural Sciences of Drexel University. So I'm still dabbling, I guess is the best way to put it, in some research and in field work, some graduate students and so forth and so on. But what I'd like to talk to you about today is really sort of looking back, particularly at the work I've done in the Devonian period, and the field work, some of the discoveries. I was trying to add up all the scientific papers and things, and we're up above 50 scientific papers, 20 new species, all sorts of additions to knowledge, and the idea that we've built collections through time that are publicly available at the Academy of Natural Sciences and will hopefully support research projects for many decades and centuries ahead, maybe, is really what I find so rewarding in the long run. But let's get started. I'm going to share screen and talk about where we're going and where we've been. Tell you a bit about the work I've been doing. So, exploration into the age of fishes, also known as the Devonian period, and the origin of limbed vertebrates, which of course is one of the very significant evolutionary innovations that occur during the Devonian period. And remember that any of these transitions aren't happening over generation or generations. It's really 10, 20, even 30 million years of slow, steady change, leading from something that we would call finned animal to something that we would call a limbed animal. So I hope we can talk about that a little bit as we go. And that's been one of the interests, is that story. But of course, the Devonian period has a diverse assemblage of animals, and I'm interested in really all of it. I'm also interested in how ecosystems are changing in the Devonian period, because ultimately it's the ecosystem change and the ecological opportunities that are driving evolutionary change. All right. So let's start just sort of with the what, who, where, when kind of thing. We are looking at vertebrates, jawed vertebrates, gnathostomes, in the group that we call bony fishes, osteichthyes. We're used to the bony fishes. If you ever fish, or if you like eating fish, we generally are dealing with the ray-finned fish, the actinopterygians. The subgroup of bony fish that I study particularly are the sarcopterygians. So those are the lobe-finned fish. So that's a big dichotomy in the history of bony fish. And within the sarcopterygians, and the only living sarcopterygians are coelacanth and lungfish, but there was a much greater diversity in the past. The group called Tetrapodomorpha are all of these forms, which essentially are more closely related to tetrapods, limbed animals, than they are to any living lobefin, which our closest living lobefin is the lungfish. So anything that is more tetrapod-like than the lungfish will fit into the Tetrapodomorpha. And that's the group that we are studying, particularly in the Devonian, to understand the origin of limbs. This is happening, as I said, late Devonian. You can see the time frame. We're looking back 390 to 365. So that's where that idea I was mentioning a minute ago. It's important that we don't see these transitions happening in the blink of an eye, even geologically. You're talking 20, 30 or more million years. Now, where? Of course, we've got to find rocks that formed in the Devonian period, sedimentary rocks that had the possibility of burying vertebrate remains with them. They have to be exposed at the Earth's surface now. Don't have any fancy way of predicting exactly where these may be in the subsurface. So we go places where the rock is exposed at the surface. And importantly, there's a lot of late Devonian sedimentary rock out there that formed in deep ocean basins or even shallow ocean basins. And those are not places where this evolutionary change that we're interested in was going on. We need to sample the ecosystems that were creating the conditions to favor the origin of limb-like appendages. And of course, like I said, all the rest of the life at the time too. So we're particularly looking for ancient stream systems. And how do we do that? Well, we get out there and we find things. Not all paleontologists have to do field work, have to go find new fossils. There's plenty of fossils in collections. But for some of these questions we've been interested in, I also just love this part of it, and I feel like we have a good team for doing it. We love to get out and find new material, go through the process of preparation, curation, and then the research and publication. So I really feel like that's how we make a big difference in the science. All right. Time is always something. So let's get the context of where the Devonian period is. It's always hard to, on a page, talk about the immense length of time of the Earth. At the bottom of the spiral, you'll see the origin of the Earth at about 4.6 billion years ago, essentially the origin of our solar system. And for the first 4 billion years or so, the Earth was cycling away, geologic processes, very simple life, unicellular life, eventually multicellular, always aquatic, so always in marine realm. So all the way into what we call the Paleozoic, which is the oldest part of that is the Cambrian period. And we know about the Cambrian explosion, kind of the diversification of multicellular life 525-ish million years ago. But even after the Cambrian explosion, everything was still confined to aquatic settings and not even freshwater settings. We get all the way up into the middle Paleozoic, and I've circled the Devonian period. So as I said, it stretches from more than 400 to about 360 million years ago. We're interested in the late part of the Devonian period. Notice something really consequential here on this diagram, which is the way green first appears in that late part of the Devonian period, actually middle and late Devonian. That's absolutely a critical sort of precondition to the origin of life on land and even the origin of life, diversification of life into stream systems. Because stream systems would have been completely barren, nothing living up in there because there really was no primary producers, nothing, coming into the water, planktonic or rotting plant material or anything until the Devonian period. So really, when you think about it, if the Earth is 4.6 billion, we're really looking at the greening of the Earth at about 400 million. So less than 10% of Earth history had there been plant life on land. And so really, this was the beginning of something very significant. And what we're looking at is, so we're looking at stream systems and we're looking at those organisms that are developing features for shallow water living and mostly predators. But we're also looking at what those plants are doing, what the primary producers are, what the detritus feeders, so these are things like armored fishes. We want to understand the food chain in the Devonian period, starting with plants and ending with things like the earliest limbed animals. All right, so I hope we've set ourselves in time. How about set ourselves in sort of an evolutionary tree? These are I mentioned gnathostomes, and so to the right, these are all gnathostomes. So your sharks, and then your bony fish, as I mentioned. Within the bony fish, osteichthyes, there's your sarcopterygian side. There's your actinopterygian side. So these are just a few examples of living groups in living forms in each of these groups. Within the sarcopterygians, yes, we get coelacanths, then lungfish. But then in the Devonian period, and let me highlight the Devonian here, you could see some really significant new additions along this tetrapodomorph lineage. Again, things that are more closely related to tetrapods, limbed animals, than they are to lungfish. So more derived on the tree. And as you can see, from tetrapods, we come to the modern groups, the amphibians, the reptiles, including birds and mammals. And of course, within this reptile lineage are things like dinosaurs. There's been so many amphibians, mammals, and things like mammals going all the way back in time. So what I want us to focus on when we're talking about the origin of limbs is this tetrapodomorph, also called stem tetrapods, just being a major branch on the tree of life. Okay? So that's where we are. So we've set ourselves in time, set ourselves in a phylogeny, a tree like this. Let's look at a little bit about, again, when this was happening. Remember I mentioned that so much of Earth history, 4 billion years of Earth history, is happening here in the pre-Cambrian. And when we get into the Paleozoic, we get the start of the diversification of multicellular life. And as far as vertebrates go, so backbone animals, boy, they start in the Cambrian very simply, very, very simply. If you saw something wriggling around today, which was like these earliest forms, you might not even notice. It might look more like a worm swimming through the water. But what happens during the Cambrian, Ordovician, Silurian, so this is about 525, all the way up to into the Devonian, are important features, just as important as limbs go when we're talking about vertebrates. So the origin of, early on down here in the Cambrian, the origin of heads, organisms that have sensory organs kind of consolidated on one end. So the origin of heads in animals. What about the origin of paired fins, meaning a set of pectorals, the front, and the pelvics? That doesn't happen until we get in through the Paleozoic. What about the origin of jaws? Jaws were such an important thing to sort of establish that what we think of as most vertebrates body plan. We do have jawless vertebrates today, things like lampreys and hagfish. Anyway, so they're very primitive. But here it is, again, this is where I'm focused, late Devonian, and that's where limbs. And again, so we can see whether it's heads or paired fins or jaws. Each of those things are kind of sequentially creating an organism that then in the late Devonian sort of had some of the raw material, if you will, for origin of limbs, for trying out some new ecological opportunities. So that's when the action is. So here we go. Where to go? You recognize Pennsylvania. You may not recognize what the colors mean. They're a geological map, essentially. These are the age of the rocks on the surface here in Pennsylvania. Of course, it's much more complex under the surface, but as I said, we can only really work on the surface. And you'll notice Devonian being this light blue, the Poconos and the Allegheny Plateau are the places we've had the most luck, particularly Allegheny Plateau. But there's a lot of Devonian rock, much of it marine. Some of you may have been to Deer Lake out here. All across Pennsylvania, you find crinoids and brachiopods and trilobites, all from the Devonian period. But again, those are the marine places. We're interested in the non-marine stuff. And as I said, that's Allegheny Plateau and Poconos to some degree. So here's our search area here in Pennsylvania. And again, I've been working out here for 30 years. And PennDOT has been our partner in this. They don't know it, but they've been our partner, unwitting partner, I should say. So we love it when they're going through sections of the Appalachians or the Allegheny Plateau. And as you can imagine, there's not much rock outcrop. There is Devonian rock all underneath this one little hillside here. This would be a museum's worth of fossils buried deeply in there. But again, we can't take it apart, but PennDOT does. And so they nipped off this corner to keep the road flat and spread out the rock across here. This is Powey's Curve on Route 15, just north of Williamsport. And we found some fantastic stuff right here. In fact, even this was one of the early discoveries, and it's a pectoral fin of a tetrapodomorph. So it's clearly more derived than what you see in a lungfish fin, etc. But it's also very much like early limbs. In the bones, you can see this drawing below, humerus, radius, ulna, some wrist elements, and what we call radials out here, but very similar to a phalange, a bone that's within the fin, that muscles attached to, that give that fin much more dexterity and strength than that whole ray fin side of fishes. We think of a ray fin fish, if you've gone fishing, if you catch a trout or a bluegill or anything like that, you see those fins as being full of thin rays, but they're not muscular really, and they don't have much control. They're really just steering, and they're just things for moving in water. Whereas these guys had muscular control of the fin, probably very much, or certainly very much for an aquatic living. But of course, we've got to go back to Gary Larson. Every scientist does, to say, when are great moments in evolution? It's when old structures are used for new functions. And so here we have the structures which are, somewhat similar to, or at least the architecture is there for what we see in a fin. But this animal was not using that fin in a limb-like way. Perhaps it was supporting itself on the bottom, a muddy, shallow bottom. Who knows? Maybe pushing, digging a little bit. I really can't predict. Somebody might look at this fossil someday and look at the range of motion and the muscle attachments and do more quantitative sort of biomechanical work. But Sauropterus was an early discovery that sort of helped us, start to assemble our knowledge of this transition from fins to limbs. This is the Red Hill site, and the most incredible site. It's in Clinton County out there. Incredible for several of the species that are known from there, but the range of species also makes it like a wonderful glimpse into those freshwater stream ecosystems. Don't try to read all this, but we have groups of armored fish, the placoderms, very primitive shark. Among the bony fish, we have some ray fin forms, but then a lot of lobe fin forms, including early tetrapod material, this Ichthyostyggalia. Along with them, we actually have arachnids and millipedes, so spider-like forms and millipedes, and a whole lot of plants. So this is a wonderful place for, getting that concept of what was the crucible? What were the ecological conditions where these earliest tetrapods were living? Animals that were right on that cusp of using those appendages strictly in the water to maybe less in the water and a little bit on land. And so they're right on that cusp, which again, didn't happen overnight. That happened over a long period of time. But here's a place where we could actually reconstruct that ecosystem. And actually, if this is basically where Philly is, down here in this corner of Pennsylvania, you see this outline of Pennsylvania. And this is a Late Devonian geography, basically. If you're out here in Ohio or in Western PA, you're out in the ocean in the Devonian. And there's some good Devonian fossils out here, including big armored fish, the placoderms like Dinichthyes. But right across, about halfway, and it built out through time across Pennsylvania, big floodplain. And as I mentioned, we're interested in those stream systems. And this would be approximately, the kinds of setting where something like Red Hill would form. Big meandering streams, flooding during some seasons, leaving mud and silt on the floodplains, plants growing on those floodplains. We see all that diversity. Just incredible to this was the crucible, places like this. These are some of the organisms, everything from lobe-finned fishes. These are big pectoral fins here, not much of the skull there. Armored fishes and plants. This is an armored fish. These are all from Red Hill. A little ray-finned fish, sort of a minnow-sized thing. And both this jaw and this shoulder girdle actually are those ichthyostegids, meaning those early, very derived lobe fins, which we call early tetrapods. So very distinctive. This was a humerus of one from out there at Red Hill. So the upper arm bone, we've never found a specimen that has like skull and jaw and fins / limbs out there at Red Hill. The depositional setting isn't quite right to preserve articulated animals. We have in other places, but even just this humerus showing muscle attachments and articulations for how the elbow would work and all that sort of thing turns out to be very important. Cover of science important. And this is sort of pulling all that Pennsylvania work, particularly Red Hill, together. This is meant to represent that early crucible, that early freshwater ecosystem with plants and forests established on land. But if we had gone back 30 million years before this, it wouldn't have been there. The plants hadn't evolved that tree-like form and so forth. There might be some low-growing material, going back through the Devonian, even back into the Silurian, but not much, not the kind of biomass represented by these forests. And forests and swamps where the water is in the plant growing zone would lead to all of this detritus, and little ray fins would eat the detritus, and placoderms are sucking up the mud and getting material, and then there's millipedes, and there's all sorts of stuff. So a food chain is set up for the first time. And lo and behold, lobe-fin fishes kind of find a predatory niche here. And some of the lobe fins that are developing more limb-like fins are finding new niches, even getting into more shallow, maybe into the more swampy habitats, maybe being able to escape the predation of some of the lobe fins, or to get to zones where those bigger lobe fins are not, eating all the good food. So they're finding the animals with more limb-like appendages are finding ponds where they're not in competition with these guys. So really an interesting and important sort of place. And actually, the last, I just have to give plants their due. This is a wonderful paper, just such a overridingly important concept. I just mentioned that sort of earlier in the Devonian, very low growing plants, very shallow rooting. That's all there was out there. Look what happens through the Devonian period. Plants get much bigger, much more tree-like. Naturally, they are sending roots much deeper into the soil. If you know the impact that plants have on creating soil, kind of recycling the rocky breakdown products that create soil, adding organic materials. Look at this. Look how important the Devonian was from early to late Devonian for the kind of nature of plants on Earth. And think about how impactful that was. Think about stream systems. Think about what roots do to stabilize stream banks and create levees, and therefore change the patterns that streams follow. And so the banks aren't always eroding. You get more meandering style rather than braided style streams. On and on. Hugely impactful. That was my little soapbox for plants. Gotta give plants their thing. Let's get back to vertebrates. So we found a lot of lobe fin forms and early tetrapods here in Pennsylvania. Remember, we're in Pennsylvania. We're sort of in the latest part of the Devonian period. A lot of these fully aquatic forms, and then early tetrapods. Clearly, there's sort of like transitional forms missing. We don't have a lot of the stepwise change that we can hypothesize happened between these forms. This is a form called Panderichthys that was known from Latvia. So clearly these types of fully aquatic lobe-finned fishes are closely related to the early tetrapods, but we really did want to find more of the transitional forms, anything that would help with that. So Neil Shubin and I hooked up. He was a professor at Penn. I was at the academy. And we talked about where else in the world could we look at Devonian rock. This old textbook of mine that I had used at Franklin and Marshall College in historical geology in probably 1979 had this map in it. And this is one of those things of, wait, let's look at this book. Wow, here we go. This map changed my life. What it's showing is on the North American continent, one of the very broadest sorts of rock types out there. And you see in the American West, there's lots of limestone, et cetera, marine deposits, same in the Midwest, muddy, silty deposits. But the Catskill Formation, which is what we do in Pennsylvania, those are those stream deposits. We knew about this Greenland stuff. Those are also stream deposits. And that had been pretty well studied. So there's the Catskill that we've been doing. There's the Greenland stuff, been studied by Danes and British folks. Similar finds to what we got in Pennsylvania. But these Arctic islands, the geologists had been there and mapped it, but no paleontologists had looked at it. So Shubin and I got it in our heads that we were going to expand our field project and go up to the high Canadian Arctic. And it was 1999 was our first trip. It was also the year that Nunavut territory formed. It was a subset of what used to be called Northwest territories. There's still some Northwest territory over here, but all of this is now called Nunavut. It's an Inuit homeland. The capital is here. So you fly from Ottawa up to the capital, Iqaluit. And from there on out is just remote. And we fly all the way out to a research base at Resolute Bay and then you can do that commercially a few times a week. Flights go up there. But then we took small planes and helicopters out to various places across here to do our field work. And it's Ellesmere Island over here where we had really all of our good finds. Here's the terrain as we head out. We're in a twin otter plane, small plane out of the Resolute Bay research base. Pack ice breaking up. A lot of people ask, do we notice any difference? And all that I've seen, and we've been up there from 1999. Last time I was up there was 2013. And I hope to go back again actually next summer. Yes, the pack ice is breaking up. We hear from the natives that it's breaking up earlier than it used to, and it changes their hunting patterns, et cetera, et cetera. But anyway, we're interested in the rocks. And as I said, luckily the geologists had mapped for us, so we determined where we want to set up camp based on the geology around us. We put ourselves in the middle of as much late Devonian stream system rock as possible. And there's a rock formation called the Fram Formation, which formed in environments quite similar to the Catskill Formation, but a bit older. We're talking back to about 375 million years rather than 365 here in Pennsylvania. So we basically get put out there a month at a time. We take all of our food, equipment, everything we're going to need because we can't get resupplied, or it would cost an awful lot of money to get resupplied. And then we walk and we prospect, we call it, where we're crossing these landscapes, looking for rocky outcrops, getting down close. It seems absurd that this is a frozen fjord in July up behind us there. This is Goose Fjord. And we find things on these blocks and you're in this huge landscape, not a person within 100 miles in any direction. And here we are with like a dental pick. Because we found pieces on the surface here, this head on the left here, from the dorsal side to the left and ventral side to the right, is an armored fish, a placoderm. And we were pulling out pieces, a few were on the surface, and we were pulling out others here. And that's what it would look like. This is one of these armored fish, Bathyrialepis, very common actually. We call it the junk fish of the Devonian. It's really everywhere, this thing. But always good to find and find good material. So this is from a helicopter as we flew into a valley where we've spent an awful lot of time now, parts of 4 field seasons. And this is all that Fram formation. You can see the strata dipping to the right side here. So earlier in the Fram, these red beds. Now I'm moving to more later in the Fram formation. In other words, between like 300 and say 80 million years and about 375 million years, the accumulation of these ancient stream deposits. Obviously, nothing like it looks like now. These would have been tropical, warm weather sorts of places. But right here, we had camped in this valley first in the year 2000, and we found a site. It was our Nunavut 17th site we had logged in the year 2000. And we found a few interesting pieces. And so we went back the next year. Actually, 2 years later was the next time we were back in the valley. So 2002, and we found more good material. And we've ended up collecting at that site because it produced the material that's so exciting. There it is right there, NV2K17. After we had dug out the fossil the beds dip, as you can see. So the fossil zone, which we originally encountered up here on this surface and with some broken pieces kind of eroding down under here, but we found a fossil zone and it's dipping under the surface. So we need to take off rock above, just like a strip mine does when they're trying to follow a seam of coal into the earth that's tilted. Here we are. We've taken off rock above now. This, I think, was 2004. And you can see these plaster jackets. The surface we're all working on here is dense with critters. And sometimes parts of critters, sometimes more complete things. But right there, that particular plaster jacket, we had just put on this here. This was 2 sides of a lower jaw and part of the snout of an animal. We don't try to prepare these. In other words, take off all the rocks and see what's there completely in the field. You couldn't do it. It falls apart. So we glue things up in the field, kind of consolidate the block, then put plaster around it to make a rigid like cocoon, flip the block and put more plaster on it. So we do end up with this like egg or cocoon with the rock and what we hope will be a great fossil inside of it. By the way, if you're seeing that, there's the bottom surface, the top surface of the skull. If it was all there, is down, and this is where the 2 jaws meet at the chin. So we were actually very excited about this because it represented a lobe-finned fish with somewhat of a flattened head. We could tell that just by what we could see here. And that's really the design that we thought these early tetrapods or transitional forms might have. My colleagues Neil Shubin here and the late Ferris Jenkins in that hole in the ground. And I just like this picture because, here we are. It's like 40 degrees, drizzly, rainy. You're digging in a hole, sitting on wet rock. You can see the tarp behind it, gathering water, trying to keep other things dry. Trying to get plaster to dry in those conditions is hard enough. But look how happy we are. Because when you're finding good fossils, everybody's happy. And that's the key. That we were at this particular place, we knew that we were getting things that were going to be interesting and important. So with just that's that same piece, the 2 sides of the lower jaw before plaster was put on it. This is Steve Gatesy from Brown University. Brought that back. We shipped back that plaster jacket. And then back in the prep lab in Philly at the Academy of Natural Sciences, we start to take the rock away. And this is all the same specimen. This was the side that was originally facing down early on in the prep. Later in the prep and later in the prep still, although a lot has been done. You can see the eyes, see this sort of flattened shovel-shaped head, shoulder girdles. We even have scales on the back of this animal. So incredibly exciting. And this has turned out to be just about the best fossil that I've ever worked on. And got to give props to Fred Mullison. He's retired now. fossil preparator who did hundreds of hours, months and months of work. This particular specimen might represent at least 4 months of work. And then, come back and do new things to see different parts of it and all the rest. That's the same specimen, somewhat more fully prepared. And you start to get the idea of the animal. And when we start, this is where we could start the research now. We could start comparing this skull and the sutures, meaning on the skull, to other forms, the shoulder girdles, which is what is here, the fin material that is in there. This, for example, is the skull. So there's the Tiktaalik skull. This is an early tetrapod from Greenland called Acanthostega. These are some other Devonian forms that are known, but not very well. So Tiktaalik really fit kind of between what was known by some of these forms and these early tetrapods. So similarities to the tetrapods and similarities to the finned animals as well. There's a 3D surface scan of that same specimen. It's called NUFV 108, Nunavut fossil vertebrate. It's cataloged, it's curated at the Canadian Museum of Nature in Ottawa. Although right now this specimen is back in Philly. It's at the Academy of Natural Sciences in this exhibit that we set up last fall that will be there through this summer. It's called Life on to Land, the Devonian, and actually it covers a lot of what I've been talking about, particularly Pennsylvania finds. But the nice people at the museum in Ottawa let us borrow back this incredible specimen, just incredible transitional form, borrow it back and put it on display at the Academy. So if you get a chance to go over there and look at it, please do. It's quite exciting. Anyway, so ventral side on the left, dorsal here. You can see we have all the shoulder girdle materials, fin bones, etc. I say fin, should I say limb? We'll talk about that in a second. I want to just show you where some of the technology that, paleontology tends to be a fairly, kind of old-fashioned-y, or at least it's had that reputation. But new technologies have really allowed for some amazing new techniques. So a grad student out in University of Chicago, which is where Neil Shubin is now, a guy named Justin Lemberg, CT scanned this entire skull, and we had always been interested in the sutures, how the different bones fit together, because they looked like there was mobility between the sutures. You see mobility in fish skulls all the time, meaning one bone slides past another during biting or air gulping or whatever it might be. We don't have mobility in our skulls. Mammals don't, but you hear about snakes and that mobility. Fishes really have a lot of mobility in their skulls, particularly for suction feeding. But anyway, watch what Justin was able to do was really just to start to play with the range of motion. So here we're looking dorsally. Look how the cheeks swing out. And we had wondered about this because of sutures we saw here. These are the hyomandibula, which turn out to be an important function, a pivot point. From below, you see the whole palate move. And so what Justin saw was very much that this skull did still have mobility, although perhaps less than some of the more primitive forms around it. And actually, sort of came up with a hypothesis of a feeding mechanism for this form, which was sort of transitional between an animal that's fully aquatic, just needs to suction feed in the water, versus an animal that needs to be able to snap down on its prey without all of that head mobility. So interesting sort of intermediate, even when it comes to the feeding mechanism. Another specimen from that same site, NV2K17, Tiktaalik, a larger individual. You might recognize two lower jaws here. So we have a whole skull here. Look at this. There's a humerus plugged right into the shoulder girdle there. This is unprepared. This is really rough. This is almost just as we got it back from the field and opened up the plaster jacket around it. Other fins. So we actually have about 50 specimens that we can identify as Tiktaalik roseae. And they're part of several hundred specimens from this same site, which represents some of the other diversity there. But I just wanted to show you that we have this wonderful fin material. Look at the 3D quality for a 375 million year old organism. You know, three-dimensional shoulder girdle all the way around, clavicle up to the clithrum, and then other bones that attach to the clithrum. So we're really able to see how these fins attach. In fact, this is where the humerus plugs in, this kind of like saddle-shaped thing here. And this is part of this is a humerus that has been pulled out of there a little bit. So what it's enabled us to do is to really kind of put together a scenario for change through this 20 to 30 million years, sort of additions of interesting features in some lineages, whereas other lineages stayed primitive. But so here's a very aquatic form, Sauropterus. Remember we found in Pennsylvania. That's the shoulder girdle. So the head would be here to the left, the fin would articulate here. And if you can imagine a fish body here, that fin is articulating just along the side of the body, kind of facing backwards here. Then we get into a form like Eusthenopteron. Similar, very aquatic, but that scapulocoracoid is getting larger, maybe allowing that fin to do more dexterity. Look at Tiktaalik. That scapulocoracoid has gotten huge, and it started to orient downwards, underneath the animal more. And the whole scapulocoracoid has shifted sort of below that other area here, the big blade. And then in something like Acanthostega, which is an early tetrapod, that scapulocoracoid is actually a significant bottom half of this element. And this is the more dermal element that we see up above here. And that articulation for the fin is actually lateral. So the orientation has changed. Its position within the shoulder girdle has changed. Let's add a humerus. So we know the humeri of these things. We all are, I'm holding my humeri here. So we have humerus here. Again, the construction, let's add the ulna and the radius. So this is showing us kind of that through this sort of addition of features leading toward early tetrapods, you start to see a real change in the orientation of that appendage. I'm even going to add what we call the radials, or down here we might call digits, like our digits. And then I'm going to add the fin rays themselves, which is part of the skin. Remember I talked about actinopterygians and how they have rods? Well, so do lobe fins, but lobe fins also have that internal skeleton, which I just went through with the humerus and radius and ulna, etc. So Sauropterus clearly had these big, long lepidotrichia, the fin rays. And so that's clearly an animal with a surface area for moving through water. And same with Eusthenopteron. Tiktaalik, interestingly, started to reduce those fin rays. They were becoming much more robust. So instead of just being like almost hair-like, not hair-like, but thin, you know, I don't know, the size of a paper clip sort of width or something. They get even thicker and stronger in something like Tiktaalik. And then when we get to early tetrapods, they're gone. No more bones within the skin of the fin. No more lepidotrichia. So many of you may be saying, well, when do we call it a fin? When do we call it a limb? Similarly, in colloquial terms, when do we call it a fish, or when do we call it a tetrapod, which is kind of a fake, which isn't really necessary because tetrapods are an outgrowth of things we call fish. But let's talk about it as fins and limbs. We arbitrarily, from what we know out there now, will put Tiktaalik within the fin side of things because it still has lepidotrichia, and we'll put Acanthostega and many other forms like it within the limbed side of things because it's lost them. That is a complete human construct. You know, it's just so we can name things, and we humans love to name things, and we feel like we want to put things in cubby holes, but nature doesn't work that way. When is it a fin? When it is a limb? If we could find every fossil out there, we wouldn't find an even dividing line. So we have made this arbitrary fin to limb with the loss of lepidotrichia. All right. Just wanted to show you, this is the same specimen of Tiktaalik. You've all seen this front end of it. There was actually more bone in a block we took out behind, and a pelvis in a little block out to the side. And some of the pelvic fin bones there too. And there's the pelvis itself, which actually shows us something really interesting. The hypothesis would have been, okay, a pelvis is small in these forms. These animals mostly were front-wheel drive using their pectoral appendages. And that's what it looked like before we found Tiktaalik. But clearly this pelvis is well developed. And so here's our lobe fin, like very aquatic type form. Look at Tiktaalik. There's the shoulder. When we add the scale of that pelvis, look at how much. So the pelvis, although the architecture hasn't changed much, its importance, its function as a kind of buttress is still there. And that's more like what we see in early tetrapods. So clearly this hind fin was becoming somewhat important for locomotion in Tiktaalik. So here we are. We've now plugged something in. We have a transitional form, but gosh, there's infinite numbers of more transitional forms, right? There's no such thing as one form, one missing link. Everything is a link between something and something else. But we're lucky to have something like Tiktaalik, which kind of splits that difference there. There's a reconstruction. Color and pattern is sort of the artist's idea of shallow water predator, using things like pickerel and muskellunge as models. So going to go back to where we started a little bit. There is that stem tetrapod group, the fish tetrapod transition happening in that group. And a couple of the take-home messages here is that the seemingly major evolutionary change, we can really start to break down into a sequence of small changes through time. Doesn't happen overnight. Mosaic evolution, meaning, hey, still had scales, but it had more limb-like structures. It had lost some of the features that we see in aquatic forms. This is something like Tiktaalik. So mosaic meaning different parts are changing at different rates. And the sequence of that change, we can't stand back and predict that. Our closest relative is a lungfish. We can look at a lungfish. We can look at something like a salamander, which is maybe most similar to the earliest tetrapods, although not necessarily related, but in body form and function. And we can look at the lungfish and the salamander alive today, and we couldn't predict the sequence of changes, much less what was causing those changes. But the fossil record allows us to sort of see those sequences, and if we're lucky, tie them to environmental change as well. And just don't use the word missing link. A transitional fossil, not the transitional or anything like that, a transitional fossil. It's only because we're so hung up on Linnaean classification, and we live with terms like fish, and we live in terms like tetrapod, and it never leaves room for the gray areas in between, and that's absolutely what we're working in here, is sort of gray areas. So we can't put everything in its cubby hole. All right, I wanted just to touch on this. I'm at time. This is flying. I have about 5 to 10 more minutes. Sorry about that. Let's keep going. This is the globe back there in the middle and late Devonian. And here's Pennsylvania to the south, and then in what we work in Nunavut, the Canadian Arctic there. So look where they are. They're equatorial on this Euro-American continent, which is North America plus parts of, and Greenland and parts of Europe and so forth. Not far. And then the other continental masses are down below. And we learned about some deposits of late Devonian, actually they're middle Devonian, down in what is now Antarctica. And you can see Antarctica was tied up with Australia, Antarctica, India, the African continent, all the rest, in this Gondwanan. So we thought, let's go there. Let's look at those deposits. Sometimes I wish we didn't do it. It's just tough. So here we are geologically. We're actually in the Gavetian, the latest Gavetian, and what's called the Aztec siltstone. And in time, we thought that would be very interesting for maybe more transitional forms. Here's the geography. Sorry, dry throat. Here's the geography. So South America is up this way. We fly to New Zealand, take a military aircraft to McMurdo Station here in McMurdo Sound. And these are the Transantarctic Mountains. And again, thanks to the intrepid geologists who had mapped out there, we knew there was rock of the right age. It formed in the right sorts of environments, and it was practically unexplored. So there's McMurdo Station, amazing place supported by National Science Foundation. Great science. We go down there in December and January. It's their summer. This is two different ships come in each year. This is actually the icebreaker. They come in from the ocean, which is way out that way. But the icebreaker comes in, opens the channel, and then a fuel ship comes in and a cargo ship comes in. And the cargo ship takes out all of the trash and all of the scientific stuff that may have been generated during that year. Magic is that the helicopters can take you out there to the field sites. Here we are flying out to our first field site. This was 2016 to 2017. Glaciers come down through these, what they call the dry valleys, but these rocks are exposed. So very dramatic, very cold, no life, hard logistically challenging. You do a lot of training. The US government, National Science Foundation doesn't want you getting stranded or falling in a crevasse or freezing to death or whatever. So there's a lot of training involved. But as you can see, we're doing similar stuff, using similar methods, just in more difficult conditions down there. This is the Aztec siltstone. And here we are splitting out rock. A lot of these lighter colored things are armored fish, placoderm elements all through here. Here's another site where we're wrapping up. Everything is carefully, so it's collected, it's wrapped in toilet paper, dated, packed away, and eventually shipped home. Here's a beautiful placoderm, two sides from this stuff. There's the same species, but tiny. So that's about a penny. That's a New Zealand penny size. Here's this form we just saw is the same as this, but this is a tiny little juvenile. Just the terrain we get to walk through is amazing out there. This is just alongside one of the glaciers that's coming off the ice cap and down into these dry valleys. All this big right from this side here, this just slab with a lot of lobe fin fish material. So we did find lobe fins, but nothing right on the transition. And this was the more recent field season that we did out to a place called Deception Glacier, where we actually flew in these twin otters that can land on the glaciers. We actually loaded snowmobiles into the twin otters, so this was logistically challenging. But spent about a month out there going from sort of area of rock outcrop to rock outcrop. Finding good stuff. This is my colleague John Long from Australia, and that's the head of an armored fish again. So what we did come home with were lots of sharks, lots of armored fish, some lobe fin fish, but nothing really close to that transition. And that's just sort of the terrain. There's actually there's a fossil right there. So you get an idea of what we do. Pack it all up. So this was about 800 pounds of fossil material. The research base put these crates together for us. It got shipped back to California across the Pacific on this cargo ship and then trucked across country. And here's that same crate sitting in our lab in Philly. And there's Fred again, starting the preparation. Here's the section of our collection area where everything is curated, cataloged. We have lots of studies, and I'm going to say the next generation as well will have lots of studies to do on what we've brought home from Antarctica. Oh, there's that cool little placoderm right there, the two sides. That might even be the juvenile one next to it. So that's really what we do. And this is the big part of making sure that the time, effort, research that we do is there for the future. And that's a big part of what museums do, of course, is make sure that that what we do can be documented and that new questions can always be asked. So that's it. So I'm ready for questions. I'm going to stop the share. I apologize for going on a bit. I guess we're just at about an hour, so reasonable. I guess if there are any questions, maybe Betsy will speak up and.
Yeah, we do have questions on the list. I thought Becky was on to offer them. Uh, I can always get started with some, and hopefully she'll pop on. Uh, Hold on a
All
second.
right, Becky should be able to
Okay,
read
I've
herself.
been pressing
she
the
is.
button so many times
Okay, I'll shut up.
that, yeah, no, that's okay. Eric stepped in because I had problems with my computer before the whole thing froze, but I
Uh-oh.
had an old laptop, so here I am. So, oh my heavens, thank you so much. Let me get my picture out here. Wes, can you put my picture back up? I guess he can't. Okay, well, I won't worry about that. You can hear me, right?
Yes.
Okay,
You should be able to start your own. Wait a minute. Yeah, you should be able to start your own video.
okay. Whoops. This is probably why my
There you go.
computer froze earlier. I think I pressed these buttons too many times, trying to hurry up. So, okay, we have several questions here. One, just a compliment that we always have from Bruce Costa. Deep respect for your work, goes without saying.
Thank
My
you.
heavens. You know, we all contribute in different ways, and you change the world with that work. So, a question from Ed Gracely. Were the early, excuse me, pollen is rough this year, isn't it?
Yeah. I'm
It's
feeling
hitting
great.
all of us. Okay. I'm going to skip Ed's for now because I scrolled up too fast. Can you comment on the rarity of creatures that have transitioned from sea to land versus the other way around? Has anything transitioned from land to sea?
Very much.
Oh, wow.
So if we're and I think it's a fascinating question to ponder. As far as we know, only one group of vertebrates has made the transition from sea to land or water to land. Because it certainly did seem to happen from freshwater aquatic settings onto land. And that's what we've been talking about. No other vertebrate groups on the planet today seem to be derived independently from some other aquatic group, fish group. It was really this lineage in the late Devonian. Whereas things that have transitioned from vertebrates that have transitioned from land to water dozens of times. All of the marine reptiles of plesiosaurs and mosasaurs and ichthyosaurs, probably several times. Whales, dolphins, things like seals and walruses and things are kind of in that transition. If you think about it ecologically, they may or may not become more dependent on the sea, but they are kind of partway along there. Lizard groups, I mean, I mentioned the mosasaurs, but other lizard groups through geologic time. So it has happened a lot the other way. And I wonder why. I wonder why it only happened once. And the best answer that I've heard or thought about is that when these lobe fins were making the transition, there seems to have been new opportunities, new resources to use in shallow freshwater settings and eventually moving into land more, probably for arthropods, invertebrates. But as soon as something else was up on land that might have wanted to eat you were vulnerable when you were in that transitional. And I don't, again, let's not make this anthropomorphic. Your species, your ecological niche just wasn't there, wasn't really available because something else was kind of keeping that from happening through time. So I think that's the best I can think about it. It's a neat question. It's a question that deserves a lot of thought. Why it's only happened once with vertebrates.
Yeah. That one was from Eric, and another one from Eric. You mentioned the farther back, I'm sorry. You mentioned the major farther back periods of time. What are your thoughts on using the term Holocene?
Holocene is good. Anthropocene is the one that's been in the news lately. Holocene is good. Holocene is I don't even know when people say it started, but I think it's post-Pleistocene. So we are in the Holocene now. The question has been, could we recognize a Anthropocene, something sort of since the basically the Industrial Revolution have we changed the Earth that much? And the geologists have just weighed in and said no, based on the criteria that they use for what these, I guess these are epochs, what these epochs require. So that's fine. It's a semantic thing. There's no doubt that we need to recognize that current times there are processes which are significantly different than they were before humans were on the scene. But so it's just nomenclatural. I'm not sure if that's where that question was going.
That's great. By the way, earlier when I was trying to unmute myself, you apologized for talking too long. It felt like 3 minutes.
Me too.
That is fine. It was all valuable. Okay, back to Ed Gracely here. Were these early tetrapods what we would consider amphibians today?
Great question. No. So we have to, they are essentially along the lineage leading to the common ancestor of living amphibians, living amniotes. So somewhere after the Devonian up into the Carboniferous period, there's a major split in those stem tetrapods at the node that we would actually call tetrapoda. And that leads to amphibians or amniotes. And amniotes are what lead to reptiles and mammals. So that split does not happen in the Devonian period. So those earliest limbed forms, I know a lot of times, and even in my talk, I call them tetrapods. And that's kind of, there's always this like, hey, we use terms like fish. Hey, we use terms like tetrapods. Let's keep small t's and small f's on those things. Whereas capital T tetrapoda is a scientific name for a node which comes later. Okay. And so that's where we would then start to recognize things as true amphibians. Otherwise, if we called these Devonian forms amphibians, then we would have to say that all of us are amphibians. And in some ways we're well, anyway, it gets complicated.
Okay.
That's a good question though, because that's a frequent confusion.
Dave Cattell, commenting on you packing all that stuff up, said it reminds me of Darwin sending home samples from the Galapagos.
Yeah.
Except he didn't have
it's logistically challenging and it's very worrisome. we said goodbye. We froze our butts off in Antarctica and worked for a month in the field, and then we just sort of pack things in a crate and say,
cross your fingers,
take care of this.
Yeah,
and I got home. So
yeah, like sending your kid off to college.
I suppose.
A little bit. Okay. Having a little trouble with the scroll bar. What paleontological I don't know if I said that word right. Paleontological questions are you and colleagues focused on now that can be addressed through studying late Devonian fossils?
Okay. Well, I'm it's boring, but it's because it's sort of the same. But I just think continuing to work on diversity. There's so much that is unknown in terms of the connections between this group or that group, whether it's the armored fishes or the rayfin fishes or whatever, or the lobefin fishes. I personally am sort of most interested in that. Now, students that I work with, that Neil Shubin works with, are asking cool questions, this sort of mechanical, functional anatomy. How did things chew? How did things bite? How did things swim? How was the pelvic appendage becoming more important versus the pectoral appendage? So there's kind of as science will always do, we ask one set of questions and then we count on people, hopefully not shooting down everything we did, but building on what we did although they can shoot it down if they feel that they must, hopefully building on with more detailed work or different questions and so forth. So I'm just going to keep adding to the resource. And I guess that's the key to me. And that's that legacy of building collections is something, as I mentioned early on, I'm really proud of. So,
as you should be.
thank you.
As you should be. So it sounds like you've retired from teaching and the
No
university,
more
but not from the
teaching at
field.
Drexel. But I still, as I said, there are a couple of grad students that I stay involved with. And I do go in, when I do go down to the academy, I'm mostly sort of doing a backlog of stuff. For example, that Antarctic material, we really do need to do some research projects on some of that. So I'm going to just sort of cleaning up backlog is how I say it.
Gotcha.
So and that's very rewarding, and I can do it on my own time, and there's really not much responsibility, and it's fun, and I get to be that sort of cranky old guy who comes in late and then leaves early and all that kind of stuff. I'm not cranky.
No, you've never come across that way. Somebody else asked, I didn't realize this. You have written books for children?
Oh gosh, forever ago there was a thing about running press was a press in Philly when I was pretty new there in Philly, and they did a something about just fossils in general and asked me to do that. So can't say I was very. It hasn't been a big push in my life, but I really do like communicating about science, and I really do like getting younger people connected and getting a better understanding of how exciting it is to do science. So I guess I don't have anything formal planned, but I will continue to interact wherever I can.
Good, excellent. So no more books planned currently.
Nope.
Okay. Well, I have to look those up because I have young people in my family that I want to push science.
Oh. Well, there's better ones out there than mine. Yeah, I mean, this is so long ago, and yeah,
Okay.
there's better things out there.
That's funny. Okay. Once again, I'm having a little bit of trouble. Okay, somebody asked, they missed the first half of the presentation. Will a recording be available later on YouTube? Yes, it will. And very likely hundreds of people over the next few weeks.
Wow.
We'll see it. Okay, I am still trying to scroll. Okay. What DNA diversity have you found from present day samples? I have to tell you, I don't even understand some of these questions. So
yeah. So I'm not the kind of person who would look at like the DNA of lungfish and then compare it to whatever different tetrapods or different ray fin forms and stuff. So I don't work on that level. Neil Shubin is very much he works with sort of modern groups and looks at the genetics of, change and anatomical change, morphological change, and then connects that to some of the, not directly, but sort of similar patterns that are seen in the fossil record. So, and DNA just doesn't last a long time. We don't get anything from the Devonian. In fact, some people talk about fragments of organic material, DNA and such, preserving for a pretty long time, but in terms of complete DNA, it goes away very quickly. So it's not really part of the fossil record, especially what I look at.
I can understand that. So we won't be cloning Tiktaalik then?
Nope, no.
Okay. Are your photos and diagrams available in books or online?
Lots of photos have been used in different ways or in different blogs. There's no one clearinghouse for any of it. So I don't know, the Academy through time has certainly used them in lots of different ways. So that's the best I can say. Some of them, some of the diagrams, I'm not sure if there's anything that was in today's talk, are some things that Neil Shubin had put together for Your Inner Fish, that book. Some of the photographs in that book are mine too, but they're not big and fancy. Anyway.
That was a very good book.
Oh, it's a great book. Fantastic.
Yeah.
Yeah. And it starts out just at that NV2K17 site. I mean, we actually went there, 2013, we went there with the film crew for Your Inner Fish, which was exciting, but awkward.
Okay, from Michael Lewitt, who also asked the DNA diversity question. Have you found fossil records of species with non-5-digited? Michael, was there another part of that?
I think I know what he's asking.
Okay, good.
Yeah, very much. I mean, personally, I haven't, things like Acanthostega. So it's not one of our finds, but it's a very well-known late Devonian form from Greenland. It is not five. Does not have 5 digits. Now, when we go back and we start talking about the lobe-fin fishes, we're not going to call them digits yet because they still have those fin rays, and they're one reason or another they may not be homologous to digits. But things like Tiktaalik, there's a big fan of those internal skeletal elements. Certainly not 5 precisely yet. So 5 is something that happens, as we get into true tetrapoda, capital T tetrapoda. And that's fascinating. And why limbed animals have all started with that design and it's so common in so many limbed animals is a really interesting question.
Okay. What is the Arctic summer like? I'm guessing it's not like a New Jersey summer.
No. It's it doesn't get below freezing, which is nice, or it gets right about down to freezing, and it will snow a little bit. So I'd say between 32 Fahrenheit and 45. When the sun is out, it's gorgeous. You can actually kind of feel warm, strip down, take a few layers off.
A few layers
when
off.
when it is windy and drizzly and cloudy, it's pretty miserable. And there are days we can't work because it's raining too hard and we can't, it's too dangerous to be crossing rivers and walking on wet rocks and all that kind of stuff. But it's light all the time, whether it's cloudy or not. But like if there's no clouds, the sun is up 24 hours. It drops a little bit lower in the sky at night, but it comes back up, goes around. But of course, that means other times of year like this, it's dark all the time. So, yeah,
that would be rough. That would be rough. I know you've done a lot of speaking. Is there a question you always expected to ask that nobody ever asked?
Well, that's an interesting question.
And maybe not.
Yeah, gosh, Becky, nothing's coming to mind. I'm sorry.
That's fine. That is a perfectly good answer.
Yeah.
Perfectly good. Okay.
I'm sure if I thought about it, I would come.
That's all right. I always think that people who do a lot of speaking, whether they're movie stars or scientists, you get a lot of the same questions. And I'm sure sometimes you think, wow, how can I answer this so it doesn't sound like I've answered it 2,000 times?
Well, this group has had some really good questions.
It's a great group.
Yeah.
It's a great group. Okay, Bruce, I see you had a very long question on there, and I was trying to figure out how to get that one out, and it looks like you've shortened it. How do you guide young science enthusiasts into a specific field of science?
Cool. Yeah, and I do hear from a lot of people interested in paleontology, of course. And actually what I often do is say, look, when you're young, get the breadth of the science. Be comfortable with all the sciences you do. You don't have to be an A student in all of them, but understand that you're building a foundation, and that if you're going to then become a paleontologist in the future, you need to have that foundation firmly established. And then when it comes to students that then continue or have already done that and are almost ready for college or something like that, I usually say, make sure you understand geologic time, geology, take some geology. You don't have to be a geology major. You can certainly come into this with sort of evolution and zoology and some biology for sure. But you need to at least have that context, the kind of stuff that I talked about early on, setting where you are in time and so forth, understanding ancient environments and change on Earth and things like that. I often tell students that are focused in paleo, hey, learn anatomy, like on Thanksgiving, save those bones, boil them, and then take them apart, and you've got a dinosaur skeleton there. So understand what makes up the front of the pectoral appendage and the pelvic appendage. What are the homologies to your skeleton? Just learn skeletal anatomy is a good one. The other one is like, be comfortable. You don't have to be an artist, but be comfortable sketching and drawing, because by drawing, you notice things, you notice the details. And so if there's a raccoon skull that you found under the front porch, sure, and people love putting that stuff out on the shelf. But if you wanted to spend a little time and sketch it and understand the shape of the head or the teeth or whatever, just being comfortable drawing is a wonderful skill to have, which helps you with observations.
Interesting. I have a great niece who, at the age of 8, in a family that is educated but not scientists, announced she was going to be I hope I'm saying the word right, entomologist. She loves bugs.
Sure.
Nobody
Excellent.
in our family encouraged this at all. But so sometimes, I looked at the question and I thought sometimes you just let them go.
Oh,
You
kids
just
are
let them.
scientists. That's the thing. Kids are naturally scientists. I'm afraid our society kind of dissuades them of that. But being curious and
especially females.
Yeah, good point.
Especially
Yeah.
females are dissuaded. So
Yeah.
Very interesting that we're all kind of saying, where did this come from?
I got somebody pulling on my leg down.
Oh.
He wanted to get up. That's Rocky.
Why are you talking and not talking to him, right?
Exactly. He was pulling on my fingers.
That's so funny. All right. I am trying to. I'm just looking to see if there's any other quick questions. Somebody in here, I don't remember the exact question now. What would you like to do next? Do you have another discovery in your mind that you would like to do, or are you about at the point where you're just looking for what turns up?
I mean, the beauty of it is I feel no pressure to make discoveries, and maybe I'm lucky that way.
Yes.
I remember in my 20s or 30s, it was like, oh, how am I going to make a name? Am I always just going to work on unimportant things? And how will I publish papers and so forth and so on? So it's very nice to not feel those sorts of, that sort of pressure. And it allows me to just, hey, on this date, I'm going to go check out my road cuts in Pennsylvania, and I may come back with something really interesting. The beauty is that, so we've found a lot of different kinds of things there here in Pennsylvania. But guess what? The next time we may find a complete skull or a more complete specimen of something we already knew about, but this will really give us more detail on that. And so all of it is very noteworthy, and all of it is something that through time just, again, builds these resources. So that's, I can't say, and that's been like the story of the work we've done. It's serendipity in large part. Like, we didn't know we sort of hoped that when we went to Nunavut and looked in the right age rocks formed in the right environments, that we would find something of the transitional ilk. But if we didn't, we would have made science out of the other stuff that we found there, as we've done. It's just that Tiktaalik has really been the flagship piece.
Yes. Yes. And you identified the best thing about retirement is the pressure's off. As I know there's tremendous pressure in academia.
It can be that way.
Tremendous. So, and Sidling Hill in the Pennsylvania Turnpike.
Yeah, that's
One
beautiful
of my
down there.
favorite areas. I grew up in Western PA, so I still go back there pretty often. And I
Yeah,
always
that's a
think
great
of you
road
when I
trip.
go through Sidling Hill.
There's even a little interpretive center there, isn't there?
Um,
I
you
think there
know,
is.
I always stop at that rest stop, but I'm
Yeah.
also focused on, let's get out to Irwin. I don't know if you've ever heard of that town, let's get out to Irwin and Clarion. And so I don't. Oh, your dog is so cute.
He's a good one.
Oh my heavens, so cute. So, don't even know how to thank you for all the work you did and the times you've come here and talked and answered questions. And we appreciate you so much.
Oh, Becky, that's really
Thank
kind of
you,
you to
Ken.
say. I appreciate everyone getting on this afternoon and listening to this. And you know what? Like I say, science is not something that's ever going to not be part of my life. So hopefully in a few years I'll have something to come back talk to you about again.
Oh, that would be great. That would be
Okay.
great. That would be great. Okay, I just have a couple announcements I want to make to people. And you live in the area, you are welcome to come. Our picnic at Fort Washington on July 14th, 11 o'clock in the morning. That is a Sunday. That's our picnic. I'll get information out on Meetup. September, we don't yet have a speaker. October, we have Robert Hicks, who's going to talk about 19th century astronomy. Sorry, not astrology, astronomy. He was supposed to come last month and he lost his voice. And then in November, Melanie Tracek King, who is a fellow of CSI. She has not decided on a topic yet, but she will be speaking. And that's what we have so far. I always look at Cuts in the Highway differently since the first time I heard you speak. Thank you so much.
And we do have April 20th. What is the harm of psychics from
Oh,
Rob Palmer, who's
thank
spoken a lot.
you.
So
Yes.
same time, third Saturday in April.
Yes.
And thank you
so much.
so much, Ted, for great enlightening talk.
I enjoy it. I
Best
really
as
did
you
enjoy it.
find new ways to promote science.
Thank you.
Okay, you can sign out.
Okay.
Thank you. Take that dog for a walk.
Yeah, that's exactly what he wants.